Our topic today is about dynamic equilibrium. This reaction is in dynamic equilibrium because products are being converted to reactants and reactants are being converted to products. Now, the water fountain I'm using as an example of dynamic equilibrium because the amount of water entering and leaving the water fountain is equal and constant, so there is no change in the amount of the water left on the water fountain. So another example of dynamic equilibrium that I'm going to perform today is using my bookshelf. So taking one book, as you see, she takes a book and replacing it with a number. It is an example of dynamic equilibrium, since there is still the same number of books placed on the shelf, even though they are different. Example of dynamic equilibrium is showing this plate of food. Now, the amount of food eaten by Kiara is the same amount of food that her friend put back on her plate. The amount of food on Kiara's plate remains the same. So this is a good example of how dynamic equilibrium works.
Hi, my name is Jasmine. I'm here to talk to you today about the types of passive transport. First, what is passive transport? Passive transport is the movement of molecules across the cell membrane without any ATP or energy required. There are three types of passive transport. The first one is diffusion. Diffusion is going from high concentration to low. Example of that would be something like a hourglass. The second one is osmosis. With osmosis, it's basically the opposite. It's going from low concentration to high. The last one is facilitated diffusion. Now, when it comes to facilitated diffusion, it is different from regular diffusion. You have things called channel proteins that actually help break down those things. Like, think of it as taking a green line. The green line later on you can connect to the red line and what other lines like the green line connects to the red line, later on the yellow, the pink, all the rest of them. But that's the way the channel proteins work and facilitate it. It has different protein channels that helps break down particular proteins. Like it may be a channel that's just for water. It may be a channel that's just for particular molecules. When I think about facilitated diffusion, I think of it like an Android charging an iPhone. Like I have an iPhone, but you can't use an Android charger to charge your iPhone. Only an iPhone charger can charge an iPhone. Or when I think about a remote, if the remote requires AAA batteries, you can't put AA. So you have these protein channels that help break down molecules such as water. The channel is only geared towards that one particular protein or ion. This what makes it easier so you won't have to use energy or ATP. This is my demonstration on osmosis. Osmosis is going from a low concentration to a high concentration. The bread represents the cell membrane. Now, I'll put water, just a little bit of water. Now, the way this represents osmosis, because it's already water inside of the bread. So like I said, the bread represents the cell membrane. So as the bread sits, the water will start to absorb into the bread. This what makes it a passive transport. It does not take any ATP or energy to do this at all. As you already see, the bread is starting to like absorb it. So it's like soggy. This what makes its osmosis. Now this is my example of diffusion, which is going from a high concentration, which is this bottle filled with water, to a low concentration, which is this empty 2 liter. So as you can see, when you flip it up, it all goes into there. And you're going into a low concentration, which is the definition of diffusion.
Hi, my name is Brianna Chemist, and the topics that I'm going to explain today are hypertonic, hypotonic, and isotonic. I will use washcloths to illustrate cells. I also use purple Kool-Aid to dye the water so you can clearly see the actions of the water. Hypertonic is when the solute concentration is greater inside the cell than out, meaning the cell loses water. I will use this washcloth soaked in Kool-Aid to demonstrate a cell that is highly saturated in the concentration. I will use clear water in this mason jar to demonstrate a solution where there is no concentration in it. Next, I will take the washcloth soaked in Kool-Aid, which represents the highly concentrated cell, and put it in a non-concentrated solution, which is the clear water. Okay. The clear water turning purple demonstrates the cell, which is a towel, losing water, which is the Kool-Aid. Hypotonic is when the solute concentration is greater on the outside of the cell rather than the inside of the cell. This clean washcloth will represent an unconcentrated cell. I will put the clean washcloth, which is the unconcentrated cell, inside the concentration, which is the purple Kool-Aid inside of this mason jar. I will let the washcloth soak in this concentration for approximately 30 minutes. As you can see, the purple Kool-Aid absorbed into the towel, turning it purple, which demonstrates the cell absorbing water. Isotonic is when the cell neither gains or loses water. The concentration is equal inside and out. Because the concentration is equal inside and outside the cell, there is no water movement across the plasma membrane. Depicted here, there is no drastic change in the color of the towel or the solute. This means the solute concentration is equal both inside and outside the cell.
Hi, my name is Chris, and I'll be discussing why cell membranes considered selectively permeable. Selectively permeable is when a cell membrane allows certain substances to pass through while others cannot. In the figure, you see a single cell membrane in the beaker. The cell membrane allows a certain substances to pass, but others were denied entrance.
Hi, my name is Shekinah Strickland. I'm in Biology 121 with the LabRats group, and I will be explaining the fluid mosaic model of the cell membrane. What is a fluid mosaic model? A fluid mosaic model is a model that describes the structure of cell membranes. It is a flexible layer made of lipid molecules with large protein molecules that act as channels through which other molecules enter and leave the cell. The fluid mosaic model also describes the structure of the plasma membrane. Within those components, it includes phospholipids, cholesterol, proteins, and carbohydrates. The barrier of the plasma membrane is also flexible. If the barrier is broken, it will regroup. The lipid molecules of the membrane is assembled in double layers. Their tail repels water while the head attracts it. So this is the tail, and this is the head. With cholesterol and carbohydrates added, it gives basic structure of a plasma membrane. Within the lipid molecules, proteins are found. Proteins are found And one of the jobs of the proteins is to transport nutrients within waste. The molecules constantly move within the structure, within the fluid change.